मेरे देख के देखते देखते
उधर रख दो, अंदर रख दे। So students, a very warm welcome to the 7th day, which is been added into twice day. So whatever I have to teach tomorrow, I will teach you today, because I might need one day to set up myself there in the new place. So I'm shifting from Delhi to new place. I will tell you about that later. And yeah, so Um, so some of the students were asking me on personal WhatsApp about writing review. If somebody has question, please write me on the side. I will answer them here now. If you have any questions related to review, reference citing, or anything else, please ask freely. So I want to give my first 10 minutes to that only. Then we can start our session. In meantime, more students will join in. If anybody has any questions related to reviews, please feel free to ask to it. Okay, literature citing. So mainly whenever you are writing your paper, it should never include any citation from Wikipedia or from any general things. It should be from PubMed always. So I can show you my paper. So when I was writing to the format of scientific reports, Nature. Okay. So in this case, this was my abstract, quite short and apt. And then I wrote my introduction. So introduction. So after the one sentence is over, so I bring the all the references them together like 5 to 7, 8, 9, 10th, 11 to 13th. So this was done automatically. If you're using a softwares like EndNote or any other software, Citavi. So I use Citavi in my case. So they will add automatically. So at the end, the references here you can see they are all from the PubMed papers. So nothing from Wikipedia or any other sources. So they are very lame, I would say, resources to use. So always use some papers, some journals which are cited in PubMed. And PubMed is your this one. So here your journals, you can write PubMed there and look for the research that you are writing the review for. So about dissertation, please ask Vatsal sir about that. And it should be structured and compact that I discussed yesterday. I don't know whether you were in the class yesterday or not. So I discussed that in quite details. about how to use your abstract, which need to be very summary of everything. Whatever you have written, it should include all the summary inside there. Or if anyone wants to be collaborate with me, so I will let you know in 1 week. So I will select some papers which seems could go in international journal, and I can pay some amount, you can pay some amount, so we can send it to some review, we can do it collaboration way, and we can publish them in international journal. But quality should be good, quality should be good. So at least like in my so at least 50 references should be there, or less will be fine also. And material and everything needs to be authenticated, very nicely done, structurally way. All the figures should have references. If you're using references from any paper, if it is your own, then it's not needed. So in my case, all the figures I have created, so I didn't use any references. And yeah, so I've created all in my papers figures on my own, so I didn't use any references figure, so I take care of that. Actually, I got a position of assistant professor in Rama University in Kanpur. So I will have a designated position from now onwards. So I could be in collaboration with some students if they are really good in writing their papers. So we can come and collaborate together from my university, Rama, and from your university, and we can publish in some of the free Web of Science journals. So we can try from these any Web of Science. If you like to publish on your own, you are most welcome. Go ahead, and we wish you all the best for the same, and make the best out of it. Okay, any more questions? I hope some of your queries might have been cleared by now. So if not, no any more questions, then we start the session. So today I will be dealing tomorrow's lecture also and today's lecture also together. So today I will be dealing with this part now from the unit 1, study of chromosome structure, morphology, number and types. So we go with the chromosome structure about today, total study about chromosome. So architecture of chromosomes in prokaryotes and eukaryotes. So basically this is the main this is the main appendix, that is prokaryote chromosome structure. We will deal with the Escherichia coli chromosomes, DNA domains, supercalling of the genomes, DNA binding proteins. And D2 chromatin structures includes chromatins, histones, nucleosomes, the role of H1 linker DNA, 30 nanometer fiber, higher order structure. Then D3 eukaryotic chromosomal structure, the mitotic chromosome, centromere, telomeres, interphase chromosomes, heterochromatin, euchromatin, DNA is one, hypersensitivity, CpG methylation, histone variants and modifications. Then we discussed a little bit about central dogma, prokaryotic gene expression and eukaryotic gene expression. So first D1, so prokaryotic chromosome structures, that is E. coli chromosomes, which is a single closed circular DNA which has around 4.6 megabase pairs. DNA is packaged into a region known as nucleoid that contains high concentration of DNA as well as proteins associated with the DNA. And there is a continuous replications and there is no cell cycle is happening. Then there are DNA domains such as observed under electron microscope, and which are as 550 to 100 domains, and they are loops per E. coli chromosomes with 50 to 100 kilobase pairs per loop. The end of the loops are constrained by binding to a structure which probably contains of proteins attached to part of the cell membrane. So they are supercoiled domains here you can see, and they are protein membrane core or scaffold in the center, and they are around 50 to 100 kilobase pairs independent loops or domains. So here we have shown 12, around 12, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 and 11, 12. Then in prokaryotes, there is supercalling of genome is happening. In E. coli, chromosome as a whole is negatively supercoiled, so they are negatively charged, and individual domains may be supercoiled independently. So direct biochemical evidence is lacking for different level of supercoiling in different domains. Then there is a prokaryote chromosome structure, that is DNA binding proteins. So histone like proteins essential for DNA packaging to stabilize and constrain the supercoiling. And then there is HU, which is a small basic dimeric protein, non-specific binding to DNA and most abundant. And there is HNS protein H1, which is neutral monomeric and partially non-specific binding. Then there are site-specific DNA binding proteins important for organization of DNA domains. And this is D2 chromatin structure, chromatin. So this is a DNA compaction. First there is a DNA compaction which then they are added to the core histones, these DNA. Then they make nucleosomes revolving around these beads. Then the histones are like molded into in parallel form. Then they make 10 nanometer beads on a string fiber, like they make a strings of fibers. Then they further scaffold proteins, they are further scaffolding and making a 30 nanometer fibers. Then further scaffolding is done, then metaphase chromosomes. These are the metaphasic chromosome structures looks like. So this is the major structure in DNA compaction. DNA nucleosome, 10 nanometer beads on a string fiber with 30 nanometer fiber and the metaphasic chromosome. Then comes mitosis. Then in the mitosis there is interphase. Then there is a prophase, prometaphase, metaphase, anaphase, and telophase. And then after the metaphase comes cytokinesis. Then comes the interphase where DNA replication is done. So in the first interphase, the chromatin is diffuse state, but in the mitosis case, it is in condensed state. You can see it here. So this is further showing the same picture how the bead string is done with the different sizes showing to you. Initially it's 2 nanometer, then 11 nanometer, 30 nanometer, 300 nanometer of scaffolding of protein, 700 nanometer, then around 1400 nanometer of your chromosome. So you can see from a chromosome structure how metaphasic condition is done, then the extended form, then the chromatin fibers of packed nucleosomes, beads on a string, then short region of DNA double helix. So like this. Then comes the chromatin structure, that is histones. The major protein components of chromatin. The four families of core histones, that is H2A, H2B, H3 and H4. An additional non-core histone H1 is added there. So here you can see there is H3 and H4, H2 and H2B. So H3 and H4 make a dimer first. Then H2 and H2B also make a dimer. Then H3 and H4 they make a tetramer. Then they make a tetramer, and this tetramer is combined with the dimer and make a histone octamer. With H2 and H2B dimer and H3 and H4 tetramer, they combine together to make histone octamer. Around this octamer, your DNA is coiled. Complete DNA is coiled. So small is 10 kilodalton of core histones, and for 23 kilodalton for H1, that is non-core. So basically they are rich in lysine and arginine and tightly bind to the DNA. So this is top view of histone octamer, H3, H4, H3, H2A, H2B, and this is DNA is coming, enters, and DNA leaves from there. This is top view, this is side view. This is D2 chromatin structure, that is nucleosomes. So nucleosome core is the basic unit of chromosome structure consisting of protein octamer containing two each of core histones with 146 base pair of DNA wrapped 1.8 times in left-handed fashion around it. And the wrapped of DNA into nucleosomes accounts for the virtually all negative supercalling in eukaryotic DNA. So this is the nucleosome. You can see here DNA in orange and histone in the blue. Then comes the D2 chromatin structure. Here the role of H1 is being shown to us. Stabilizes the point at which DNA enters and leaves the nucleosome's core. So this is how the nucleosome looks like. Linker histone H1 and H5 family. So you have 8 histone core with H1 histone here and DNA being coiled around. So C tail of H1 stabilizes the DNA between nucleosome cores. So there are some nucleosome cores. So C tail of H1 histone, that's playing the major role between the nucleosome cores here. So this is histone octamer with DNA revolving around it with 8.6 nanometer as a diameter and length is around 2.8. And there is a histone H1 being here on the outside, which is letting the two superhelical turns to be done. Again showing us a nucleosome with H1 histone H1, how DNA is covering it, how additional DNA required to make up 200 base pairs of nucleosome repeats. Then comes the D2 chromatin structure, that is the 30 nanometer fiber. So here you can see a 30 nanometer fiber with has a diameter of 30 nanometer diameter with histone H1 nucleosome linker DNA with average of 55 base pairs. So this is a high ordered left-handed helix, 6 nucleosomes per protein. So axis of coil. So this is how looks like. So chromatin structure in the higher order structure looks like this, that we have seen in previously, with the chromosomal loops we have with 30 nanometer fiber and diameter of length of 300 nanometers. So mitotic chromosome, now this is the eukaryotic chromosome. So this is sister chromatid, centromere in the center, telomere on the top, and each sister chromatids are called chromatids. So in the center, if we see from the top, this is the nuclear matrix looks like, and on the side is a loop of 30 nanometer fiber. And this is a section being carried out. So this is mitotic chromosome at metaphase. This is how mitotic chromosomes looks like. So we have dyed them, and the spindle formation is done. So these chromosomes looks like this. Simple chromosomes, mitotic chromosomes. Then this is the centromere, which is the two chromatids are joined together, where the mitotic spindle and Y kinetochores, and this is centromere of DNA. So first one whole figure, this one is chromatid, in the center is centromere, and third one is the short arm on the top, and below is the long arm, and total it's of 0.2 to 20 micrometer of length. Then specialized DNA sequences which form the end of linear DNA of eukaryotic chromosomes. So they contain up to 100 copies of short repeated sequences in humans and synthesized by the enzyme telomerase, independent of normal DNA replication. The telomeric DNA forms a special secondary structure to protect the chromosomal ends from degradation. The structure of parallel quadruplexes that can be formed by human telomeric DNA. So image is created by this enzyme here. So there are also interphase chromosomes. Interphase chromosomes, they adopt much more diffuse structure, although the chromosomal lobes remain attached to the nuclear matrix, and they cannot be visualized individually. And human chromosomes are gray in color, capped by the telomere white. So this is white telomeres on the top, and this is the gray chromosome. This is interphase chromosomes, looks like this. Then there's heterochromatin and euchromatin. Heterochromatin is the highly condensed material which transcriptionally inactive, can be repeated satellite DNA close to centromeres, sometimes say whole chromosomes. Example, 1X chromosome in mammals. And then there's a euchromatin, chromatin other than the heterochromatins, more diffuse and not visible, regions where transcriptions takes place, not homogeneous, only a portion of euchromatin is transcriptionally active, where 30 nanometer fiber has been dissociated to beads on a string structure, and part of these regions may be depleted of nucleosomes. This is animal and plant cells both have this material called uh, heterochromatin and euchromatin. So material that is present outside like this material is known as heterochromatin, and euchromatin is the inner material. So what is DNA's one hypersensitivity? So it is an active region of chromatin. Around 30 nanometer of fiber is interrupted by binding of specific proteins to the DNA. So there is some material in the chromosome which is specifically specific to the specific proteins attaching to the DNA, and they are characterized by hypersensitivity to the deoxyribonuclease. So they are quite sensitive to the DNase 1, they get attached to it, and these reasons they are called as DNase 1 hypersensitivity. So here we can see that euchromatin showing active and inactive regions, and the sites of DNase 1 hypersensitivity indicated by large and small arrows. Then there is a CpG methylation. Methylation of C5 in cytosine base of 5-CG occurs in mammalian cells, signaling the appropriate level of chromosomal packing at the sites of expressed genes. CpG methylation is associated with the transcriptionally inactive regions of chromatins. Islands of unmethylated CpG are coincident with the region of DNase 1 hypersensitivity, and they are responsible for epigenetic and may also to the RNA silencing also. So they are CpG islands and promoter of housekeeping gene. So the one with this with the dot, they are methylated CpG, and the one others are unmethylated CpGs. The next topic, histone variants and modifications. So they are the major mechanism of the condensing and decondensing of the chromatin, operate directly through the histone proteins which carry out packaging. So histones undergo post-translational modifications which alter their interaction with the DNA and nuclear proteins. So H3 and H4 histones have long tails, can be covalently modified, and these modifications could include methylation, acetylation, phosphorylations, and lead to various diseases in the case of humans. And the core of histones H2 and H3 can be modified, whereas the histone modifications act in diverse biological processes such as gene regulation, DNA repair, chromosome condensation. Now comes the last topic, that is flow of genetic information, the central dogma. So you have your DNA, from DNA it's make mRNA, from mRNA the proteins are made. So how it is done? Before it could go ahead, DNA could also do the DNA self-replication with the help of DNA polymerase enzyme. Then further with the mRNA, it could be done with the help of RNA polymerase, which is known as transcription. And mRNA could also go back to DNA with the help of reverse transcriptase. This is known as reverse transcription. So there is a sense RNA and antisense RNA. So RNA could replicate to RNA also with the RNA dependent RNA polymerase enzyme. Then translation could happen where RNA is making proteins with the help of ribosomes and transfer RNA, and leading to the production of polypeptide chain of amino acids. Showing the same thing. So in prokaryotes, how it is done? So you have your promoter regions, transcribed regions, and terminator region. So during the transcription, RNA polymerase is added. So you have a start codon AUG. Then there are stop codons, start codon and stop codons, and again start codon and stop codons. So with the help of translation, they will produce in the ribosomes and amino acid transferase, they will produce various proteins. So in the case of eukaryotic genes, first what will happen, pre-mRNA is made, then mRNA, then proteins. So there is thing called splicing or capping of mRNA is happening. So there are regions in the mRNA which are coded and non-coding. So in the exons are the non-coded regions which will be removed, while the splicing region, so that you have mainly the functional mRNA is present. Then again with the help of translation, ribosomes, amino acid transferase, tRNA, your proteins are produced. This we have done the cell organelles yesterday, were mitochondria, chloroplast, RER, Golgi apparatus, lysosome, cytoskeleton in details already. So that's it, the architecture of chromosome. Students, I also have one lectures from the same topic, but they are more subjective. Somebody wants to read in more details, you can go through this. This is from my previous. So you can go through them also if you like. They have a lot of text there, so I didn't want to go through that, but I have explained you everything about it inside that. So just the karyotyping and staining we will do, ideograms we will do now at the end of the topic. So I will share. I have this topic. We have to do this mitosis and meiosis. This we already did in the practical part, but I also have some extra notes for you. So just go through them. So we are done with the unit 1 then. So this is more of the subjective part. If you want to read more about it, you can be my guest and go through it. Yeah. So basic principles I have explained you. These are more in detail manners other chapters. So I will share 3 files with you now. So it will keep uploading, no worries. At the moment I cannot change the video quality. It will be, just try to make it 480p. So at 480p you can see things easily. Make it to 480p. Then it will be fine. So we are almost done with the unit 1 part. Now we will start with the unit 2, that is DNA and its structure, functions, types and mode of replications and repair. So we are almost, so mainly 3 parts are left from unit 1, that is CIB technique, mitosis, oh sorry, karyotype and ideogram. These three topics are left, but most of the unit 1 is complete by today. Now we will deal with today DNA and RNA. Though I have to deal with one topic today only, but I will go through them. So this is yeah, DNA. So anything about DNA? Can anyone tell me about DNA, please? What is DNA? What is RNA? What are their functions? Any idea? Yes, it's a unit of inheritance. It is, it is. Deoxyribonucleic acid. True. RNAs, ribonucleic acids, RNAs. So that's the basic difference between these two, DNA and RNA. And we will go through quite in details now about each of the types, their functions, mode of replications and repair. It's not required to go through so much information. So basically, discovery of DNA is a prime genetic molecule carrying all the hereditary information within the chromosomes, and it has the knowledge of structure would that reveal how DNA carries the messages from replications to do identical copies of themselves. So several research groups in 40s and 50s they work in United States and Europe cooperative and regularly also to understand the atoms of DNA that are linked together by covalent bonds and resulting into a 3D dimensional space. But surprisingly, it was feared that DNA might have very complicated and perhaps bizarre structures that differed radically from one gene to another. So, great leap was done by the fund to get to know about the DNA was found out to be a double helix structure. And it told us that the genes have roughly the same three-dimensional forms and difference between two genes resides in the order and the number of their nucleotide buildings blocks along the complementary strands. So, what is DNA? Many scientists worked with the exploration of DNA way back in 1868. So, almost a century before a Nobel Prize winner was awarded to Watson, Crick, and Wilkins. They were the young physicians, Swiss physicians named Friedrich Miescher, isolated something no one had ever seen, that is nuclei from the cells. So he called them as nuclein. So today the nucleic acid, that is Na, is in DNA and RNA. So they have two types of nucleic acids called as deoxyribonucleic acids and ribonucleic acids. So 2 years earlier, Czech monk Gregor Mendel has finished a series of experiments with peas, and his whatever he have discovered, they were closely connected to the finding of the nucleins. And Mendel was able to show that certain traits in the peas, such as the shape, color, was inherited in different packages. So these packages now they are called as genes. And in 1944, the American scientist Oswald Avery managed to transfer the ability to cause diseases from one strain bacteria to another. Then solving the puzzle. So in the late 1940s, the member of the scientific community were aware that DNA was most likely the molecule of life. Even though many were skeptical since it was so simple, They also knew that DNA included different amounts of 4 bases, that is adenine, thymine, guanine, and cytosine, ATGC, but nobody has slightest idea of what the molecular might look like. So in order to solve that structure, couple of distinct species of information needed to put together. So one information was phosphate backbone was outside, bases were inside, and the molecule was double helix. So on the basis of that, uh, so they discovered that the two strands are running in opposite direction, and molecule had a base pairing of the two parts. So this is how the puzzle was solved out. Then in 1951, the 23 years old biologist James Watson traveled from the United States to work with Francis Crick, which is an English physicist at the University of Cambridge. Cambridge was already using the process of X-ray crystallography to the study of the structure of protein molecules. So together, Watson and Crick used X-ray crystallography data produced by Rosalind Franklin and Maurice Wilkins at the King's College London to decipher the DNA structure. So what they discovered, first they discovered that is made up of subunits which scientists call nucleotides, and each nucleotide is made up of sugar, phosphate, and a base. And there are 4 different bases in DNA molecules, that is adenine, cytosine, guanine, and thymine. And number of purine bases will be equal to the number of pyrimidine bases, and number of adenine bases will be equal to the number of thymine bases, and the number of guanine bases will be equal to the number of cytosine bases. So basic structure of DNA molecule is helical, with the base being a stack on top of each other. So components of DNA. So DNA is a polymer which has nucleotides, and these nucleotides act as a polynucleotides, and each nucleotide consists of 5 carbon sugar. Sugar is there, a nitrogen containing base attached to the sugar, and the phosphate groups. So there are 4 different types of nucleotides found in DNA, differing only in the nitrogen space. So the 4 nucleotides are: A is for adenine, G is for guanine, C is for cytosine, T is for thymine. And these are the purine bases, adenine and guanine, your 2 ring members, 6 members, and one 5 ring membered. So the 9 atoms that make up the fused rings, 5 carbon and 4 nitrogen, are numbered 1 to 9 here, you can see, and all ring atoms lie in the same plane. Whereas cysteines and thymines, they are pyrimidines. Their 6 atoms are numbered 1 to 6, and like purines, all pyrimidines ring atoms lie in the same plane also. Then comes the deoxyribose sugar, which has 5 carbons and 3 oxygens, and we number them like carbon 1, 1 prime, 1 prime, 2 prime, 3 prime, 4 prime, 5 prime, to distinguish between the number of atoms of purines and pyrimidine rings. So hydroxyl group on the 5 prime and 3 prime carbon link to the phosphate groups to form the DNA backbone, and deoxyribose lacks on the hydroxyl group on the 2 positions when they compared to ribose and the sugar components of RNA. So the various components of DNA, nucleoside is one of the 4 DNA bases covalently attached to C1 position of sugar, and sugar is deoxynucleosides is 2 deoxyribose, whereas sugar in the ribonucleosides is ribose, and nucleosides differ from the nucleotides in the lack of phosphate groups. So 4 different nucleoside DNA are deoxyadenosine, deoxyguanosine, deoxycytosine, and thymidine. So they doesn't have phosphate group attached to them, and your deoxy Da and Dg, they are there's an N-glycoside bond between the sugar 1 and N of the purine, N9 of the purine. So there is a glycoside bond, glycosidic bonds. And nucleotide is a nucleotide with 1 or more phosphate groups covalently attached to 3' N and 5' hydroxyl groups. So DNA backbone is a polymer with an alternative sugar phosphate sequence. The deoxyribose sugars are joined at both 3' hydroxyl and 5' hydroxyl group to the phosphate groups in ester links. which is also known as phosphodiester bonds. So example of DNA backbones are 5' D CGAAT, like this. So this is how your phosphate group, sugar, phosphate, sugar, phosphate, sugar, phosphate, and to each sugar your DNA, your nitrogenous base pair is attached. Guanine, cytosine, adenine, adenine, thymine, like this. At the end, there's a 3' hydroxyl sugar group is there. At the top, 5' phosphate group is there. And each sugar is being attached by phosphodiester links between the deoxyribose sugars. And these are your deoxyribose sugars here. Then features of 5' end structure. So there is alternative backbone of the deoxyribose and phosphodiester groups. Chain has directions from 5' to 3' from top to bottom. Oxygen of phosphates are polar and negatively charged, and A, G, C, T bases can extend from the chain and stack atop each other. Bases are hydrophobic in their end. This is that DNA double helix structures looks like. So DNA is normally double-stranded macromolecules. Two polynucleotide chains are held together by weak thermodynamic force, forms a DNA molecule. So this is the structure of DNA double helix, goes like. So so the both, like what we saw single chain in previous slides. So here we have double, they come together in front of each other and they make this parallel way of doing it. So we are done with this part. So you have this again, you have hydrogen bonds, so G and C making triple bonds, A and A and T is making double bond. You have a sugar phosphate backbone on the side, and they are one helix structure is around, one helical turn is 34 angstrom, that is 10.5 base pairs, and the diameter is around 20 angstrom, that is 2 nanometer. Any questions, students, so far? Yeah, there's a this in science is always happening. They, even for me in my research, I worked for 3 years for my research, and I was found out that somebody was already working within my neighboring group from past 5 years, and they publish it before my publishing has to be done, and they got to know about my whole data in the presentations, and they got it. So this is called scooping, we call in the science. This is the dirty, I should not have told you, but this happens mostly to the PhD students or even to the big professors. Scooping, that is your whole work is gone by somebody, because all researches in the world, they are being done at very high level, and this is not sad. It's all about how you are being competitive, how efficiently you can do your work. But what I did was, so I didn't meant, I was disheartened, but I tried to change my story a little bit and make an opposite story of that, so that my work could be also published. So it should not be that my all work should go in vain. So you have to be smart in science. That's very necessary in order to be uh, a step ahead of everyone. So it's like a chess where you have to think every step just before what others are thinking, so that your all pieces should be saved. And at the end, your king is saved with the queen also. So it's like a chess, this academia. This is what I preserve at the end of it. If you don't play smart, if you don't play scientifically sound good, there are alligators, crocodiles, just waiting to eat your stuff. Sorry to use these lines, but there are also good people who will help you to go to the top of your carrier, but mostly be safe and be aware, and do not trust anyone just straightforwardly. Um, so these are the things that have to be taken care. This is what I learned from science. And but if you are positive, like this happens in every field, it's not in the science. If somebody is going to Bollywood for being an actor, or in modeling career, or making a fast food chain, or any career that you will face in the future, so you have to be very competitive, smart, and quick witted to be, in order to get the success of things, in order to win in every field. If you got in back and laid down with the failures, then it will be no, a career will be very difficult to go ahead. So this thing happen in every field, it's happening in every field, not in the science only. So DNA has two polynucleotide strands wound around each other. Backbone of each consists of alternating deoxyribose and phosphate groups. Phosphate groups bonded to the 5' carbon atom of one deoxyribose is covalently bonded to the 3' carbon to the next. The two strands are antiparallel, that is, one strand runs to the 5' to 3', while the other runs from 3' to 5'. And the DNA strands are assembled into the 5' to 3' direction. By convention, we read them the same way. And purines and pyrimidines attached to each deoxyribose projects in towards the axis of the helix. And each base forms hydrogen bonds with one direction opposite to it, forming base pairs. So we have discussed about the diameter, length, how much is the wider, major groove, minor groove. So this we have discussed. So yeah, now comes the functions of DNA. So DNA carries the genetic information of cell, consists of thousands of genes. Each gene serves as a recipe on how to build a protein molecule. And protein perform important tasks for the cell functions or serve as building blocks. The flow of information from genes determine the protein compositions and thereby the functions of the cell. And DNA is situated in the nucleus, organized into chromosomes, and every cell must uh, contain the genetic informations, and the DNA is therefore duplicated before the cell divides. That is replication. So when proteins are needed, the corresponding genes are transcribed into RNA. And RNA is first processed so that the non-coding paths are moved and is then transported out to the nucleus. So outside the nucleus, the proteins are built based upon the code in the RNA. That is translation. So based on their classifications, DNA could be classified on various based on number of base pair per turn, coiling pattern, location, structure, nucleotide sequence, and number of strands. So there could be 6 different bases on that DNA could be divided. So first is a number of base pairs per turn, depending upon the nucleotide base per turn of the helix, tilt of the base pair, and humidity of the samples. So they are in different forms, that is A, B, C, and D. Then based on the coiling pattern, according to the coiling pattern, on the basis of coiling pattern of the helix DNA is of two types, that is right-handed and left-handed. Most of the DNA molecules are right-handed, that is coiling of the helix in the right directions. So it is called positive coiling, and all the 4 forms, that is A, B, C, D, are right-handed, and there is exception to that, that is zDNA, which is left-handed double helical structure. And this DNA is considered to be associated with the gene regulation. Then according to locations, they could be of 3 types: chromosomal DNA, cytoplasm DNA, and promiscuous DNA. Chromosomal DNA is found in chromosomes and are called as chromosomal DNA or nuclear DNA. And cytoplasmic DNA is found in cytoplasm, especially in the mitochondria and chloroplast. and such DNA plays an important role in the cytoplasmic inheritance and has a circular structure. Whereas the promiscuous DNA, some DNA fragments with the common base sequence are found in chloroplast, mitochondria, and nucleus. This suggests that some DNA sequence move from one organelle to other. Such DNA is referred to as promiscuous DNA. And whereas your RNA structure, it contains ribose sugars, nitrogenous bases, phosphate groups, and instead of thymine, it has uracil. And instead of deoxyribose, it has ribose sugar, and here adenine pair with the uracil. So we have discussed that A, B, C are the right-handed, Z is the left-handed, and B is the major form that is found in the cell. Z was initially found at high salt concentrations, but cellular environment is actually low salt environment in their case. So this is the difference showing you about DNA and RNA. We have discussed them already. So how the modes of replications are in DNA? So it is of three types: dispersive, conservative, and semi-conservative. So in dispersive replications, the old DNA molecule would break into several pieces and each fragment would replicate an old and new segments, would recombine randomly to yield progeny DNA molecules. So each progeny molecule would have both old and new segments along its length. So according to the conservative scheme, the two newly synthesized strands would associate to form one double helix, while the two old strands would remain together as one double helix. In contrast, in the semi-conservative mode, DNA replications, each newly synthesized DNA would remain associated with the old strand against which it was synthesized. Thus, each progeny DNA molecule would consist of one old and newly synthesized strand. So this is about semi-conservative replications, which was postulated by Watson and Crick, along with the double helix structure of DNA. So it says that there's a progressive separation of two strands of DNA molecules are happening. Complementary base pairing of bases located in the single-stranded regions. There's formation of phosphodiester linkage between neighboring deoxyribonucleotides, and this ensures that the base sequence of newly sequenced are strictly complementary to those of the old strands. So in the DNA replications, you have one parental and there is one new strand is formed. So it consists of several steps. First, initiation of replications. First, replication is initiated with the help of cutting the replication site with the help of enzyme endonuclease. Then unwinding of strands. The two strands of DNA double helix unwinds. The opening of DNA strands takes place with the help of DNA unwinding protein. Then formation of RNA primers. Synthesis of RNA primer is essential for initiation of DNA synthesis. And then RNA primer is synthesized by the DNA template near the origin with the help of special types of RNA polymerase. Then synthesis of DNA on primer. After formation of RNA primer, DNA synthesis starts on the RNA primer. Deoxyribonucleotides are added to the 3' end and position of the RNA primer. And then there will be some short pieces happening while doing the DNA synthesis, which are known as Okazaki fragments. Then DNA polymerase degrades the RNA primer 1. This enzyme also catalyzes the synthesis of short DNA segments to replace the prime. And then these enzymes are also catalyzes the synthesis of short DNA segments to replace the prime end. And then there is a union of Okazaki fragments with the help of a DNA ligase enzyme, which could lead to the point of origin in a one direction. Then now comes the next topic, that is DNA repair system. So there are damages to the genetic material and DNA are taken care of the DNA repair systems, the various damages to DNA may be grouped into following two types. One are single base changes and structural distortions. In the single base changes, such changes which affect a single base of DNA molecule and represented by conversion of one base to another. So like deaminations of 5-methylcysteine cytosine results in thymine. As a result, these base does not pair properly with its partner base. Then comes the structural distortions. These changes generally adverse affect the replications or transcription of the affected DNA molecule, and they are represented by a single strand nick, removal of base covalent links between the base in the same or in the opposite strands. For example, pyrimidine dimers, addition of bulky adducts to base which may distort the configuration of the double helix. So the pair systems recognize a variety of changes in DNA to initiate action. So there could be of various types of DNA damages, and they will be grouped together into general categories. So direct repair, excision repair, mismatch repair, tolerance systems, retrieval systems. In the direct repair, there is a reversal or simple removal of the damage to the DNA, which is known as a direct repair or removal of the covalent bonds between the 2,4 and 2,5 carbon of the two thymine residues participating in the formation of thymine dimers. So thymine dimers are generally formed due to the UV radiations, interfeather replications and translations. And enzyme can bind to the thymine dimers in the dark but requires energy from blue light for removal of the covalent bonds between the T residues. That is why this process is known as photoactivation. Then comes excision repair. In this excision repair, damage or mispaired segments of DNA strand is excised and new stretch of DNA is synthesized in its place. So they are recognition and incisions, excisions and synthesis. So recognition and incision is the damaged section of strand recognized by an endonuclease, and this enzyme then cuts the affected strands on both the sides of damage. And excision is the after the incisions, if 5' to 3' exonuclease digests away the damaged or mispaired sections, this generates a single stranded regions in a double DNA double helix. Then comes the synthesis. In this step, single stranded regions produced by excision serve as a template for DNA polymerase, which synthesizes the replacement for the excised segment. And DNA ligase then seals the nick that remains after synthesis of the replacement of the excised section. There is a mismatch repair system where a single base pair DNAs are mismatched due to the alteration in the existent bases or due to the errors during the replication. Structure distortion results in the DNA double helix. Then comes the tolerance system. These systems deal with the damages that block normal replications at the damaged site, possibly by permitting the replications of the damaged sites, possibly with high frequency of errors. Then comes the retrieval system. These systems are also known as post replications or recombination repairs. So thank you, students. That was something about DNA structure, functions, modes of replications and repair in short and nutshell. So let's take a break here for 5 minutes. So it's 6 or 10, 7 or 10. So we meet at 7:15. Then we start with the next topic, your last topic of today, RNA structures and functions and its type. So this is the next topic of yours, RNA structures, function and its type. So majorly, yeah, we discussed in 5 minutes then, students. Just give me 5 minutes break. So I'm back. So let's finish the last topic of today. So RNA, its structure, function, and its types. With the discovery of molecular structure of nucleotide units, each nucleotide unit consists of nitrogenous base, ribose, and phosphate. So RNA is a type of molecule that consists of long chain of RNA that is transcribed from the DNA by enzyme called RNA polymerase. It is generally further processed by other enzymes. So RNA is central to the synthesis of proteins, and here a type of RNA called messenger RNA carries information from DNA to structure called ribosomes. And these ribosomes are made from proteins and ribosomal RNA, which comes together to form molecular machines that can read messenger RNA and translate the information they carry into proteins. So most cellular RNA is single stranded, although some viruses have double stranded RNA. And single stranded RNA is folded upon itself either entirely or in certain regions. In the folded regions, the majority of the bases are complementary and joined by the hydrogen bonds. And this helps in the stability of the molecule. The folded region of the bases have no complements. Because of this, RNA does not have purines pyrimidine equality and is found in DNA. This is the usual structure of one base pair of RNA looks like. You have ribose sugar, then ribose sugar is here, then you have phosphate group attached to it, phosphate group, then ribose sugar, and one on the one prime carbon atom, you have a nitrogenous base pair attached to it. So chemically, structure of RNA could be of two types, stem loop and hairpin. Stem loop look like this and the hairpin looks like this. So there are more unusual bases in RNA than in DNA, and all normal RNA chains either starts with adenine or guanine, and three types of cellular RNA have been distinguished. That is messenger RNA, ribosomal RNA, or soluble RNA. So ribosomal and transfer RNA, they comprise about 98% of all RNA, and all three forms of RNA are made on DNA templates. And transfer RNA and messenger RNA are synthesized on DNA templates of chromosomes, while ribosomal RNA is derived from the nucleolar DNA. So the three types of RNA that are synthesized during different stages in early development. Most of the RNA synthesized during cleavage is mRNA, and synthesis of transfer RNA occurs at the end of cleavage, or ribosomal RNA synthesis begins during gastrulation. So that's some comparisons shown to you, DNA and RNA. Just go through them. Then comes the ribosomal RNA first, which name suggests they're found in the ribosomes, and it comprises about 80% of total RNA. And they're complementary to the region of DNA where it is synthesized. And in eukaryotic, ribosomes are formed on the nucleolus. And the ribosomal RNA is formed from only small sections of the DNA molecule. Hence, there is no definite base relationship between ribosomal RNA and DNA as a whole. So ribosomal RNA consists of single-stranded twisted upon itself in some regions, and it has helical regions connected by intervening single-stranded regions. And sometimes their base pairs are complementary and they are formed by the hydrogen bonds in the ribosomal RNA. So like this, sometimes they're making hydrogen bonds and sometimes they are single-handedly like this. So ribosomal RNA does not show purine pyrimidine equality and stands unfolds upon heating and refold upon cooling. And ribosomal RNA is stable for at least 2 generations, and the ribosome consists of proteins and RNA. So 70S ribosomes of prokaryote consist of 37S subunits and 50S subunit. And 30S subunit contains 60S ribosomal RNA, while the 50S subunit contains 23S and 5S ribosomal RNA. An 80S eukaryotic ribosome consists of 40S and 60S subunit. In vertebrates, 40S subunits contains 80S ribosomal RNA, while the 60S subunit contains 28, 29, 5.8, 5S RNA. So there are 3 types of ribosomal RNA, that is high molecular weight ribosomal RNA with molecular weight over 21S to 29S, and there is a high molecular weight with a below a million, and then there is a low molecular weight RNA around 58 RNA ribosomal RNA. So this is how the structure of ribosomal RNA looks like. So you have 40S, 60S, and then this is mRNA is attached here. This is small subunit, this is large subunit, okay. And then in the center is your transfer RNA, and here's your anticodons are attached, then codons are attached with the groove attached to it. In between there's a groove with the polypeptide chains of amino acids being produced in this tunnel, and at the bottom is the endoplasmic reticulum. So Jacob and Monod proposed the name messenger RNA of the RNA carrying information of protein synthesis from DNA to the sites of protein formation, and it consists of 3 to 5 prime 5% of total cellular RNA. So size of messenger RNA is about 500,000 and sedimentation coefficient is around 8s, and they contains large quantities of messenger RNA stability, and this is because messenger RNA, like other RNA, is constantly undergoing breakdown. So mRNA has the following structure features. It has cap, non-coding regions, NC1, initiation codes, and coding regions. So it has 4 major paths. So cap region is the 5' end of the messenger RNA. In most eukaryotic and animal viruses, molecules found as a cap. Then as a non-coding region, this cap is followed by a region of 10 to 100 nucleotides regions, which is in A and U residues and does not translate protein. Then comes the initiation code, that AUG in both prokaryotes and eukaryotes, and the coding region consists of 1500 nucleotides on the average and translate proteins to made up to 73 to 93 nucleotides. So messenger RNA, it was formerly thought to have 20 transfer RNA molecular types exist, one for each amino acids. It has been but recently shown that several cases that at least 2 types of transfer RNA for each amino acids. So there's some exceptions to it. Then comes the transfer RNA, and also known as soluble RNA, sRNA, or tRNA as a transfer RNA. So after ribosomal RNA, the second most common RNA in the cells is transfer RNA. It's also called as soluble RNA because it is too small to be precipitated by ultracentrifugation at 100 G. And it constitutes about 10 to 20% of total RNA of the cell, and transfer RNA is relatively small RNA having molecular weight of about 25,000 to 30,000. A nucleotide sequence of tRNA was first out by Holly et al. And yeast L90 transfer RNA. Since then, sequence of about 75 different transfer RNA ranging from bacteria to mammals have been established. So this is a L-shaped structure which has D arm, variable arm, and anticodon arm. So there are 3 different arms are present, and there's a T-psi arm and a septus stem. So like this, you have your complete transfer RNA structures looks like. So the 2 ends of molecules form a septus stem regions where the amino acid is attached, An anticodon is exposed single-stranded regions in a loop at the end of anticodon arm. So two other stem loop structures are named after the modified nucleotides that are found in those parts of the molecule. So there is a D arm, contains dihydrourylite residues, while the T-C arm contains ribothiamidylate residues. Then there is a pseudo-urodylate residues and cytidylate residues in that order. And tRNA is usually drawn in the cloverleaf form to emphasize the base structure in the secondary structure. The cloverleaf form, the structure is. So tRNA could be of three types: class 1, 2, and 3, with name as D4V45, DSV45, D3VN. This means that D stem has 4 base pairs and 4 to 5 base pairs in the variable loop. Second class 3 base pairs in D stem and 4 to 5 base pairs in the variable loop. And D stem class 3 base pair in the D stem and large variable arm at the end. So starting amino acid in eukaryotic protein synthesis is methionine, while in the prokaryotes it is N-formyl methionine. And tRNA molecule specific for these two amino acids are methionine tRNA and N-formyl methionine tRNA respectively. So tRNAs are called as initiator tRNAs and they initiate protein synthesis. And initiated tRNA of prokaryotes, eukaryotes also have some difference. So initiated tRNA in prokaryotes for initiation of protein synthesis, we have tRNAF met, and for eukaryotes tRNA met. And prokaryotes, Holobacterium sutriburium is however reported in initiate protein synthesis with tRNA met and has an AU base pair at the end of accept or stem. So D loop of prokaryotes initiator of tRNAs contain A11 U24 base pairs, and other tRNAs have Y11 R24 base pairs. So they are quite specific. These transfer RNAs are, that is, two important steps in translations during the protein synthesis are activation of amino acids and transfer of amino acids to tRNA. So each amino acid has specific activating enzyme, that is tRNA amino acyl synthetase, and there are 20 different tRNA amino acyl synthetases for the 20 common amino acids found in proteins. So some tRNA synthetases can activate more than one amino acids, that is, they show only limited substrate specificity, and thus isoleucine tRNA synthetase can also activate L-valine and valine. So enzymes, however, recognize only a specific set of tRNA, that is L-isoleucine tRNA synthetase, and recognize only tRNA, ALU and valine tRNA synthetase. So thus specificity is involved at two stages: activation of the amino acids and transfer of the amino acids to tRNA. So that's it from the RNA side. So that's it, students. So I have taken your tomorrow class also. So if I'm not available tomorrow, I will take class day after tomorrow, but I will definitely take your class to finish this course off. So we are basically left with with the 2, 3 and 4 part. So we are done with, we have to finish second and third 4 part, and we will do it in the next in 2 days. So we have 2 days left. So tomorrow will be class or not class, I'm not sure, but if there's class I will send the link. Okay, so I have to leave for my next class. I have very less time. So thank you very much, and I see you day after tomorrow then. If not tomorrow, then day after tomorrow. Hope I see you also tomorrow. Bye bye. Take care. Stay safe. Wear mask. Sanitize your hands. Clean your hands with soap. Don't move outside a lot. Okay? Then take care. Bye bye. Wish you all the best.